and scientist, Rajiv Gandhi Center for Biotechnology. He will enlighten us with the latest trends in post-targeted approaches for newer antivirals and disease management strategies. Now, let me welcome our dynamic president, Mr. Rajesh Jha, to this webinar. I now welcome Dr. Sreekumar, the speaker of today's topic, who is a renowned expert in the area of immunization. I also welcome our very active secretary, Mr. A. Sunil Kumar, and MC member, Mr. K. George Koshi. I welcome one and all who are attending this webinar. Thank you.
Thank you, sir. It is on the absence of President, I am straight away going to introduce our guest of the day, Dr. Sreekumar. It's a very important topic today. Mrs. Dr. Sreekumar, sir, is going to tell about the pandemic. The pandemic has shaken the whole world. But not only the management community, it has shaken the whole world, and we are very eager to hear from you, sir, about the vaccines and the recovery from this big pandemic of this century. So, coming to the introduction part of Sreekumar, sir, Dr. Sreekumar currently heads the molecular virology laboratory in the Rajiv Gandhi Center for Biotechnology. He completed his master's degree in immunology from the Indian Veterinary Research Institute, Izzat Nagar, Bareilly, UP in 1996 with a gold medal, and then worked for a short stint as a veterinary surgeon in the Department of Animal Husbandry, Government of Kerala. Subsequently, he joined the Animal Biotechnology Laboratory at Rajiv Gandhi Center for Biotechnology as research scholar and obtained a PhD from the University of Kerala in the year 2004. Later, he was recruited as a faculty in the center in the same year in 2004 and established the molecular virology program on emerging viruses, focusing on chikungunya and dengue in 2016. His laboratory played a key role in initiating molecular diagnostic services in Kerala during the explosive outbreak of the diseases in the past decade. As we all know, we have been the state has been affected very much with chikungunya and dengue during the past decade. Research conducted by his laboratory identified newer mutant strains of these viruses circulating in Kerala, transovarian transmission of chikungunya viruses in mosquitoes, and multiple serotype co-infections in dengue patients. He received the Fulbright Nehru Professional and Academic Excellence Fellowship in 2015 and worked as a visiting faculty during the period 2015 and 2016 in laboratory of Professor Diane Griffin at the Department of Microbiology and Immunology, John Hopkins Bloomberg School of Public Health, Baltimore, Maryland, USA, wherein he elucidated the critical role of mutations in chikungunya virus NS3 protein macrodomain in affecting the virulence. In his 17 years career as a faculty in RGCB, he has mentored 8 PhD students and has carried out 7 projects funded by external funding agencies. He has authored and published 42 manuscripts in national and international journals of repute, including Nature Communications, Proceedings of National Academy of Science, Journal of General Virology, and Journal of Neurovirology. He is a regular speaker in conferences and delivers radio talks and appears in visual media for creating awareness on emerging viral infection. His current research focuses on understanding host pathogen interactions in SARS-CoV-2, dengue, and chikungunya virus infections with a goal to develop host-targeted approaches for newer antivirals and disease management strategies. On these lines, the studies in these laboratories have elucidated critical host molecules that play a role in restricting chikungunya dengue viruses and also key signaling molecules in dengue virus-induced vascular permeability that results in shock syndrome. Thank you, sir. Thank you for accepting our request and and for enlightening us over the next 1 year, over the next 1 hour, about the disease and the vaccines. Welcome you, sir. Now I welcome you to the board, sir, on to the talk. Thank you, sir.
Good afternoon. First of all, let me thank the TMA for giving me an opportunity to talk. And also, when I was asked to make a presentation, this was always a confusion, like whether I should talk first on COVID and about the disease, or on how to control, or about the vaccines. So finally, I thought it will be good to talk a little bit on the vaccines, because that is the in thing now. Everyone is worried whether to take the vaccine, how efficacious it is. And also, even though we listen to this about the vaccines for long, most of us don't know about the background of vaccines, and maybe how it works, and whether it is good to take a vaccine, and those areas. So I thought, let us have a discussion on this, and maybe I will make a brief presentation, and later on we can take it into a discussion mode, so that we can have some thoughts shared and discuss on that. So I'll just give a short presentation, maybe 30 minutes or maybe 40 minutes. Then we'll go back to the details. My screen is visible to all? Yes. So
it is visible, doctor.
Okay, thank you. So the title is Vaccines Post-COVID-19 Towards Newgen Immunization. So as all of us know, the vaccination is one of the great public health achievements of human history. And we know about the smallpox vaccination. And on May 14, 1796, Dr. Edward Jenner, he inoculated an 8-year-old boy named James Phipps with the secretions or the secretions from a pustule of a cowpox victim. And later on, he found that that inoculation has helped to prevent smallpox in the individual. And as we have apprehensions about the vaccines now, even during that time, a lot of apprehensions were there about these vaccines. And this is a cartoon that I could get from the internet, where they show those vaccinated individuals start developing maybe features like cow, because the vaccine source was originated from cowpox. So, but later on, you can see, maybe the first vaccine was against smallpox, but the next vaccine came after almost 90 years later, when Dr. Louis Pasteur identified a successful vaccination strategy against rabies. We know that rabies is a deadly disease, and even now we don't have any other treatment to save a patient who is showing symptoms of rabies. Only option is a preventive vaccination. And one good thing about rabies is this is one of the diseases where we can take a vaccination after knowing that we are exposed to the virus, like getting a dog bite. But the development of rabies vaccine was very, I would say, a serendipitous event. He was very, ingenious. Ingenuity was shown in the development of this vaccine, because Dr. Pasteur, he was inoculating rabbits with the rabies virus, taking out the spinal cord, drying it in the sun, and later on inoculating the dried material into the patient. This is Joseph Meister. On July 6th, 1885, this first inoculation was done on Joseph Meister, who was exposed to or who was bitten by a rabid dog. And after the continuous inoculations for 14 days with this dried material, this dried material was also like dried up to different levels of dryness for different days duration. And finally, this Joseph Meister was the first patient or first person who was saved after getting bitten by a rabid dog. And this was history. And even now, we celebrate the July 6th as the World Zoonosis Day. Even when this happened after 90 years of the first vaccination identified, even by Edward Jenner. Now, the impact of vaccination in the history of diseases or in the history of medicine has been tremendous. And we see that we have vaccines for a large number of diseases, and a significant reduction in the number of cases has happened after the introduction of the vaccination. As you can see in the slide, there has been almost a very significant reduction in the number of major diseases like diphtheria, polio, measles, pertussis, Haemophilus influenza infection, and meningococcal infections. And polio is on the verge of eradication. We know that smallpox has been eradicated. So this has, when you look at the vaccination history, in 1900, there were 2 human virus vaccines available against 2 human virus vaccines available against smallpox and rabies. And there was only 3 bacterial vaccines against typhoid, cholera, and plague. And now, if you look at the number of vaccines, there are now over 20 vaccines, vaccine preventable diseases. Vaccines are available for almost all these infections. Some of the vaccines are not that successful, but vaccines are available. Now, the major vaccine covered diseases like tuberculosis, polio, hepatitis B, measles, diphtheria, inactivated polio vaccines, influenza virus vaccine, rubella vaccine, measles vaccine. And you can see that the coverage has almost more than most of these diseases, the vaccination coverage is more than 50%. And many of the diseases like yellow fever is still localized in certain parts of certain geographical regions in the world. So the vaccination coverage, we cannot say that it will reach a global coverage, but travel restrictions are there to those areas where yellow fever is prevalent. And so those people who are traveling to those areas need to take the yellow fever vaccination. Rotavirus vaccine is also mostly, it is in the developing world, this is being taken. And so slowly this vaccine coverage is going up significantly. Now, the most interesting thing about this vaccine is the time lapse between the inception of a vaccine program or development of a vaccine program and the time by which it gets licensed. So you can see there are major diseases, still many of them have no vaccines available. For example, we have this malaria, tuberculosis, then dengue, then Ebola. These are some of the vaccine diseases with the vaccines being developed aggressively. But still, we don't have a successful vaccine for any of these diseases. And among all these, the oldest would be malaria and tuberculosis. You can see even from 1880 or even 1882 onwards, we are trying to develop a vaccine for these diseases. So for most of the diseases, this green line shows the licensed vaccine is available. And you can see that the successful vaccine has been made. And the most interesting part of this whole story is the time duration that we needed to make a vaccine against COVID-19. See, the smallest duration, like even in within a time frame of less than 1 year, we could generate a successful vaccine for COVID. See, that depends on the disease that we are dealing with. Fortunately, we don't see too much of antigenic variations in the COVID virus. But at the same time, we look at the dengue virus. There are 4 serotypes of the virus, each of them behaving like one particular organism or one particular or unique organism. So we need 4 different vaccines to prevent one disease called dengue. And there was a vaccine which was developed by Sanofi Pasteur, which was which had undergone clinical trial in Philippines. And later on, they found that this particular dengue vaccine was not successful to prevent dengue by certain serotypes. So even though it was preventing infection by 1 or 2 of the dengue serotypes, it was not able to prevent the infection by the other 2 serotypes. And even those individuals who got vaccinated, they had problems, and now there is a litigation going against the Sanofi Pasteur against the dengue vaccine. So this vaccine is always
Hello, sir. Sir. Hello.
Sunil, I think the connection has gone.
Connection. Some. We will wait. Actually, I think connection of Sreekumar sir got disconnected. We will wait for his.
Sunil, contact him over phone and find out what is the status.
That is what we were doing. Actually, his connection got disconnected. He will be joining.
sorry, there was some problem with the network.
Sir, okay, sir. We can start, sir. No problem.
So shall I continue?
Yes, sir. Yes, sir. You
Please
can
go
continue.
ahead. Please go ahead.
Please go ahead.
So I was just mentioning about the Gavi, where it's a vaccine alliance among the countries involved in the group, and it will help in supporting the knowledge and even the financial assistance for developing vaccines. Now, why are the vaccines so special? We know that the vaccines promote health. Unlike many other health interventions, they help healthy people to stay healthy. That is very important. And removing a major obstacle to human development. Vaccines have an expansive reach. They protect individuals, communities, and entire populations. And the vaccines have rapid impact. The impact of most vaccines on communities and populations is almost immediate. Even now, we know that after the introduction of this COVID vaccination, now yesterday, I think as per the today's number in India, almost 2 crores of people have been already vaccinated. And we see that in many areas in the world, after the introduction of the COVID vaccine, there is significant reduction in the number of new cases of COVID. And also, like between 2000 and 2008, vaccination reduced global deaths from measles by about 78%. So the vaccines have very rapid impact in reducing the disease. Then vaccines save lives and costs. Recently, a panel of distinguished economists put expanded immunization coverage for children in 4th place on a list of 30 cost-effective ways of advancing global welfare. So there are major programs in the world which is supporting development of new vaccines. And before I go into the details on COVID-related vaccines, let us have a brief look on this host defenses on which these vaccines are looking at. We have this intrinsic immune system, innate immune system, and adaptive immune system to prevent an infection from a pathogen. So the intrinsic immune system is our physical and chemical systems, that is the skin, mucous membrane, tears, low pH, and surface cleansing actions of the mucus. All this comes as the intrinsic immune system, which is a very non-specific kind of protection that we get from the pathogens. Then there is an innate immune system, which is the first line of immune defense. Most of the time, what happens is when a pathogen is entering into our system, within the cells or in that vicinity, what is the kind of immune defense that is happening. So we also call it as a cellular innate immune defense system. So, but that is also very non-specific in the sense it does not differentiate between the pathogens, whether it is tuberculosis bacteria or whether it is a virus or whether it is a fungus. This kind of immune system will be behaving almost in a similar way. It will not differentiate between the pathogens. Then we have the adaptive immune system, which is tailored to the pathogen. So as this cartoon shows, the intrinsic immune system has a low threshold of protection. Innate immune system little more, and the maximum protection is given by the acquired immunity. And this is the acquired immunity that is what is being targeted by the vaccination. So what happens is whenever this organism is, or a vaccine or a virus is entering into the cell, into the system, there is a local replication happening, and this is taken to the lymph node in the body, and later on all the immune reactions happens within the lymph node, and the products of the immune response, that is the antibodies or cell-mediated immune response, it reaches the circulatory system from where it reaches all parts of the body and gives us a protection. And the immune response to a vaccine, if you see what happens is this vaccine is introduced, and I can explain it in detail if someone is interested, but I'm just giving a very brief overview about the system that is happening when a vaccine antigen is introduced in the body. It is taken up by certain cells called as the dendritic cells, and these dendritic cells present the antigen to the vaccine antigen to T helper cells and also T cytotoxic cells. And this T helper cell will stimulate the B cells, and it will lead to the production of the plasma cells and the production of antibodies. And this is what we usually talk about, the antibody mediated immune response. And these antibodies is the one that we measure in our blood to see whether we are protected against a vaccination. But in parallel, there is also another arm of the immune response that is working especially against viral antigens, where a cytotoxic lymphocyte component is also involved. And this is very important for providing protection against intracellular pathogens like viruses and certain bacteria which proliferate within the cell. So essentially, to have a complete protection against an invading pathogen, we need to have both the components of the immune system, that is the antibody mediated, these are the antibodies shown, these plasma cells are the one that is secreting the antibodies, and there will be a memory B cell population, and this memory B cell population gives us a long-term memory response, and this is supported by the T helper cells that is We also call them as memory T cells, a fraction of them. And there is a cytotoxic T lymphocytes. So these cell-mediated immune response plus the antibody-mediated immune response gives us the complete spectrum of protection against infectious diseases. And an ideal vaccine should be the one which will provide us the activation of both these immune responses. And this is very important for, as I said, intracellular pathogens like viruses, including the COVID vaccine. Suppose we want to get a COVID vaccine, an ideal COVID vaccine, it should stimulate both arms of the immune response. And we would have listened to some people telling that who have recovered from COVID infection don't have sufficient antibody responses. Maybe because they don't generate sufficient antibody that is detectable in the system. But even in that case also, they might not get another COVID infection because they will have a supporting cell-mediated immune response, which will be giving them an additional protection in the absence of the sufficient level of the antibody response. So that happens in a natural infection. But in case of vaccination, we have to tailor design these vaccines in such a way that it stimulates both arms of the immune response. Now, the most important function or the principle behind vaccination is the immune memory and specificity of the adaptive immune response. For example, when I give a vaccine against antigen A, there will be a primary and immune response against this particular antigen. And when I give a booster dose, this the immune response against the antigen A becomes highly activated. So we get a significant boosted level of the immunity against the particular antigen. Then this antigen, anti-A response is not sufficient to give protection against antigen B. So for that, we need to give another injection for antigen B. So that is the kind of specificity with respect to the vaccination. So the immune response is only against the antigen that we are giving. The another most important aspect of the immune response is the lag time. That is because of the memory. So the first antigen when we give, that is the first vaccination or first infection when we get, the peak immune response takes a little longer time than when it happens during the second time. So this is because of the memory of the immune response. And this is the most important parameter that helps us in getting resistance to new second time infection. And that is why in most of the time in vaccinations, we cannot stop with one vaccine. And to heighten the immune response and also to make the memory response strong, we need to give a booster dose also for all these vaccinations. Even in COVID vaccine, you would know that there needs to be 2 vaccinations taken at a distinct time apart to get the sufficient immune response. Now, what is the advantage of having vaccination? See, natural infection will give us an immunity, but natural infection will lead to the disease. So we have to endure the illness and we have to undergo the full course of the disease, including the complications of the disease, which may sometimes, if it is a severe disease, that will cost the life for the individual. And that is the cost of a natural infection, which can be prevented by vaccination. And both this natural infection and vaccination will give us the same kind of an immunity. Then second thing is, all these vaccines are designed in such a way that it does not induce any serious adverse reactions. So the complications caused by vaccinations are much less than the natural infection. So that is why we prefer a vaccination over a natural infection. I have, I came across many of my friends who were telling, see, in younger individuals, this COVID is mild, and why should we get a vaccination? So we cannot predict the course of COVID infection in younger or older individuals. There are so many unknown parameters, but we know that the vaccination against COVID produces only very mild symptoms, or maybe a febrile, a mild febrile condition lasting maybe less than a day. And the complications that can come because of a natural infection is much more unpredictable than a reaction to a vaccination. So that is why always this vaccination is advisable. Now, the safety, vaccine safety is a very important parameter. Like other pharmaceutical products, vaccines are not completely risk-free. See, this is one thing that we need to accept. So most of the people think that vaccines are risk-free and it will be 100% safe. But just like any pharmaceutical products, vaccines are not risk-free, and adverse event will occasionally result from vaccination. Although most adverse events are minor, like redness at the injection site or mild fever, more serious reactions like seizures, anaphylaxis can occur, but a very, very low frequency. And the main problem or main thing we should remember about the vaccine is given to a healthy person who is, no one will recommend a vaccine to be given to a person who has some comorbidity or some disease or immune suppressing conditions. So it is always given to a healthy person. So the expectation to the safety standard is much higher for vaccines than for the medicines that is given to sick people. So that is why in all the countries, wherever these vaccines are being practiced, there are regulatory authorities who are constantly monitoring this vaccine response. And even for COVID, all these individuals who are getting COVID vaccines have to be registered, and they are being followed up, and they are being asked for any adverse event, so that this can be corrected in due course, and even that can lead to the withdrawal of the license for these vaccines if it is a severe complication. The adverse events against vaccines can be classified, or it can be classified into various categories. So it can be the vaccine product related reaction. For example, the technology that is being used for developing the vaccines. When we have the Covaxin, which is an inactivated vaccine, we have Covishield, which is a vector based vaccine. We have Mrna Vaccine, which is an mRNA based vaccine. So all these vaccines use different technologies. So if this particular technology has a problem, sometimes this can lead to vaccine related adverse events. And sometimes it can be vaccine quality defect related reaction. Maybe a particular batch of the quality control was not proper, and a particular batch of the vaccine had a problem. So that can lead to a reaction. Then immunization error related reaction, the timing of the immunization, or the age group, or if there is any problem in the immunization part, it can lead to the reaction towards vaccine. Immunization anxiety related reaction. This is most commonly observed in all the people, and all are worried about getting a vaccine because they are all otherwise healthy. And when an immunization is done, they have the anxiety towards this vaccine. And that is one of the most common mild adverse events that is observed against this vaccine. Then very rarely you can see coincidental event which is unrelated to the vaccine as such, but it might be a coincidence that some persons have severe events that is happening because of a vaccination. So if this will be extremely rare even, but sometimes it also occur in a mass immunization program. So there has to be a balance between the efficacy of the vaccine and also the vaccine associated risk in any of these vaccinations. And what we are planning to achieve in a vaccination. So in a vaccination, one of the primary criteria is the individual should be protected against the disease. But along with that, the major readout for a vaccination success, a vaccination program success, will be the herd immunity. See, these are three scenarios which is depicted here. One is a situation where there is no vaccination. For example, in the beginning of the COVID outbreak, you see that this brown indication is a person who is infected, who has a disease, and all the blue ones are people who are susceptible. And there is no vaccination in the community, so the disease very easily spread in the population, and you can see that only very few people are left unaffected. So this is the normal scenario that would have happened in case of the COVID outbreak if we never had any restrictions or any lockdowns or any preventive measures taken during the initial phase of the COVID. Now, when we start doing a vaccination and the coverage is below the threshold for the herd protection, what happens? See, you can see that in the community, there will be two kinds of individuals or maybe three kinds of individuals. One is a person who is having a disease, and there are a group of people who are partially, who are immunized, and there are people who are susceptible. So when this diseased individual comes into contact with a vaccinated individual who is immune, there is no transmission of the disease. But there is another channel that is open, like when this diseased individual is coming in contact with a non-immune person, he becomes diseased, he can transmit the disease. So a partial coverage of the vaccination will not give a protection to the population. So even when we start with this COVID vaccination, unless we have sufficient coverage, whatever we have done so far will become waste. So that is why this vaccination program has to reach to large number of people, so that we should get sufficient coverage or herd immunity to protect the spread of the disease. Now, when this vaccine coverage is above the threshold for the herd protection, what happens is majority of the individuals are immune. And even if there is one individual who is having disease in the population, his chance of getting into contact with the susceptible individual is significantly minimized. And we see that even though there are few number of people who are not vaccinated in the community, there is a threshold of protection because of the herd immunity. And this is the ideal condition that we want to achieve. And even with any vaccination programs, especially for these explosive diseases which can cause explosive outbreaks or pandemics, we cannot achieve 100% vaccination coverage, but that is not important, that is not required. So this is the herd immunity threshold for vaccine preventable diseases. For example, you will see that measles, we need to have around 90 to 95% herd immunity threshold for getting a protection in the population. That is why we need to do an aggressive measles vaccine campaign to prevent or to establish the herd immunity. And essentially, this is directly proportional to the basic reproduction number of that particular pathogen. This basic reproduction number, in simple terms, you can mean that one infected individual, from that particular individual, how many individuals in the vicinity can get infection. For example, from a measles infected person, maybe 12 to 18 people around him can get the infection. That is the very high reproduction number. So when you take the influenza virus, which is also another airborne disease, the basic reproduction number is much less. It is 1.5 to 1.8. So when we look at the herd immunity threshold that is required for measles, we say that we need around 95% of herd immunity threshold. At the same time, for influenza, we need only around 33 to 44% of herd immunity. So it is significantly low. So even though both the diseases are airborne diseases, there is, because based on the character of this virus that is causing the disease, the herd immunity threshold varies because the basic reproduction number varies. So if this is the case, let us just assume what could be the herd immunity threshold that is required for SARS-CoV-2. Just see, SARS-CoV-2 is also a droplet infection like influenza, but influenza can spread through air. But SARS-CoV-2 need a direct droplet contact. So it is still lesser, spreading potential, transmission potential than influenza. So the basic reproduction number is average. It is between 1 and 3, maybe on an average around 2 or 2.5. And so when you can, look at the chart, you can see that around 50 to 60% of coverage is required to get a sufficient herd immunity for the preventing SARS-CoV-2 from the community. So our target in the community will be not to vaccinate all the Indian population or all the world population, but at least 50 to 60% of the population when they become immune to the disease. It can be either by the active vaccination or by the natural disease that has already spread in the community. We will see a significant or great decline in the SARS-CoV-2 spread, and we may see that after some time this disease will not be in the form of an epidemic. It will be only an endemic disease which is localized to certain pockets in the country. So this is what we want to achieve in vaccination program. Now, vaccines against SARS-CoV-2. So what is the basic principle that has been used in making these vaccines? So most of you would have heard about the spike protein and the shape of this virus. And this is the most important protein in the virus that is permitting the virus to enter into the lung cells or into any cell that it's want to infect. And we also have heard about this particular protein called ACE2, angiotensin converting enzyme 2. This is the receptor for this virus. This is present on the surface of the cell, and this spike protein will bind to the receptor, and it will mediate the infection. So whenever there is an ACE2 antibody or anti-spike antibody present in the system, it will block this. And this is what happens. This ACE2 and this is the spike. And in the spike protein, there is a receptor binding domain, that is a RBD. It is a red one. This is engaging with the ACE2 receptor, and this binding will help the virus to enter into the cell. So whenever there is an antibody, this antibody will engage the receptor binding domain, and this antibody receptor bind, this complex will prevent this virus binding to the host cell. So this is a very straightforward mechanism. But the problem is, whenever these viruses are very smart, and because of their evolutionary nature, whenever this kind of neutralization happens, the new viruses that is emerging from the few infected cells which are non-neutralized, which are infected by the non-neutralized virus, they will keep on varying their receptor binding domain. So you will see that there are variants coming up. These are some of the new variants of the SARS-CoV-2. We have heard about the Brazil variant. We have heard about the UK variant. We have heard about the South African variants that is circulating. And we even have quarantined people who have visited UK, South Africa, and all that. To prevent the spread of these variants. The problem is, whenever these variants are coming, unfortunately, if the variation is happening in the receptor binding domain, this red region, this antibody will not be, even for the vaccinated individuals, this antibody will not be able to bind to this, and there will not be any neutralization happening. See, we have two kinds of antibodies. One antibody that is directly binding to the receptor binding domain, we call them as the neutralization antibody, neutralizing antibody. And there are antibodies which will bind to any portion of the spike protein, maybe this gray regions of the spike protein. And this will not neutralize, or this will not prevent an infection. So we call them as non-neutralizing antibody. So we actually, after a vaccination, what we want is a neutralizing antibody. And this antibody has to bind to this particular region. Then only we will see that the person is effectively protected. So when we make a vaccine, we need to ensure that this particular vaccine will protect us against all the variants that can emerge. So it is not very easy for an RNA viruses like coronavirus, or maybe chikungunya virus, or dengue virus, or influenza virus, where these viruses keeps on mutating. And there will be new emerging variants that is coming up, which may be vaccine escape mutants. So even in COVID vaccination, that is one point that we need to remember. If we slow down the vaccination, if we take again another 1 year for completing this vaccination process to cover the herd immunity, we will see that some of these viruses will be vaccine escape mutant, which will start spreading, and which will start creating a second wave of COVID infection. So one of the major things that we need to be careful is to complete the 50% or 60% threshold of vaccination, herd immunity, reaching the herd immunity level as early as possible by augmenting the vaccination effort, increasing the number of vaccine centers, and increasing the coverage of the vaccine among the population. Now, in the case of SARS-CoV-2, we have seen that there was an accelerated vaccine development that had happened. For example, a usual traditional vaccine development start with the design, the exploratory stage, then the process development, preclinical toxicological studies, phase 1, phase 2, phase 3, which will take anywhere between 5 to 7 years, and the approval phase and everything. By the time the vaccines are manufactured, it takes around 15 years or longer. What we have seen in case of COVID is, since we had a prior information or prior experience with the original Hong Kong SARS-CoV-2, and also with the Middle East respiratory syndrome coronavirus, we had an idea what is the ideal target for developing a vaccine. For example, the spike protein is the best protein to make antibodies against. And when all the process for the preclinical development and toxicology, which was almost completed for the initial SARS coronavirus, but since that infection died down after maybe causing around 8,000 infections across the world, these vaccines were never developed, but the technology was in place. So immediately after this new virus emerged, we were fortunate to use these technologies to immediately develop the product that can directly go into the clinical trial. And then what happened was there was a telescoping of all these phase 1, phase 2, and phase 3 trials that was done, which could be completed in months' time. That is what we have seen now. And within, maybe in 10 months' time or less than a year's time, we have the vaccine made. And in parallel, what was happening is while the phase, immediately after the phase 1 trial itself, the pharmaceutical companies, or especially the Serum Institute and Bharat Biotech, they started manufacturing these vaccines in large doses. And they were doing it at risk. They were hoping that phase 2 and phase 3 trials will go without any issues. And finally, the same thing happened. There was no serious adverse reactions against these vaccine. So they could get the approval. And now we have a successful vaccine that is available to administer to the population in less than a year of the onset of the pandemic. So this is where we could do wonder during the present crisis. Now, see, all these clinical phase trials, all the currently available vaccines have undergone all the clinical trials. The only, the handicap with these kind of trials is even after the phase 3 clinical trial, the detection of adverse events, especially the rare adverse events, cannot be done with this phase 3 trials. And for that, we need to continuously monitor these vaccines in the population. Maybe it can be called as the phase 4 kind of evaluation. So only after administering the vaccine in large populations, we will be able to know whether there are any rare adverse events happening because of this vaccine. But otherwise, all the common events, adverse events, can be monitored through all these phase 1, 2, and 3 clinical trials, which all these the vaccines currently in use have qualified successfully. Now, when we started with this COVID pandemic, there were large number of vaccines that was that was in development, and even some of these vaccines have completed all their 3 phase trials, and it is being manufactured and used in other countries, like for example Mrna Vaccine and all these Sputnik vaccine. All these vaccines are being used in other countries, but in India we have 2 vaccines that is being currently used. So how effective are these COVID vaccines? And there have been phase 3 clinical trial results coming up. Some of these vaccines are claimed 95% immune response, neutralizing immune response. The initial studies with the Oxford vaccine was around 70%, but now the latest studies with the new vaccines, the Oxford vaccine is claiming around 82.4% the efficacy. The Bharat Biotech vaccine, that is the Covaxin, is claiming around 80% efficacy. And the Covaxin trial was released very recently. It was published on March 8th. And they say that it is been protecting around 81%, it is showing around 81% efficacy. See, what is the major difference between these 2 vaccines? Most of us know about it, but for those people, for those of you who does not have an idea about the difference between these 2 vaccines, let me just briefly tell. This Covaxin is an inactivated vaccine. What you do is you grow the virus in a culture in large quantities, and it is inactivate. The total preparation, to the total preparation, we add inactivating agents like beta-propiolactone or maybe sometimes formaldehyde. But in this case, I understand that it is BPL, beta-propiolactone. And this will kill the virus, inactivate the virus, and this whole preparation is used as the vaccine. And that means it will have the spike protein in it. It will have other viral structural protein in it, and also the components of the cell culture also present in this vaccine. And this is one of the classical methods of vaccine preparation, and does not need sophisticated systems, but it is much faster than any other vaccines, because already especially in India, that the technology for making classical vaccines are already in place. So that is why Bharat Biotech could directly go into an inactivated vaccine platform and make the vaccine. Whereas the Covishield is a new generation vaccine. See, I have shown two technologies that is used in the new generation vaccines. One is the replication defective virus vector DNA vaccine, and second is an mRNA vaccine. So this Covishield belongs to this category. This has adenovirus, chimpanzee adenovirus as the vector. That means the outer shell is formed by the chimpanzee adenovirus, and inside that there is a replication defective DNA of the adenovirus, into which a gene of the spike protein, the coronavirus spike DNA, is incorporated. And once this vaccine is injected into a patient, this DNA is injected into the host, enters into the host cell through the adenovirus, and this DNA get into the host cell nucleus. mRNA is formed. This mRNA will make the protein, and this protein will be presented to the immune cell, and this will make antibody against the coronavirus spike protein. And this will, as I said earlier, this will protect the individual. This spike protein antibody will protect the individual from the COVID infection. Now, in the case of mRNA vaccine, instead of having an outer shell of a virus vector, it uses a liposome, and inside that, instead of a DNA, it has an RNA. So it is a liposome particle into which an mRNA is trapped, and this mRNA will be injected. And this mRNA need not go into the host cell nucleus. In the cytoplasm itself, it can make protein, and it can evoke an immune response. So essentially, both these vaccine, one is using a DNA virus as a delivery system. Here it is using a liposome as a delivery system. And essentially, it will make intracellular proteins, and it will be presenting to the immune response. The main advantage of this vaccine is it will be making intracellular proteins, and these proteins are presented to the cells of the immune system. So it mimics a natural infection. So it will evoke, as I said in the beginning, both an antibody mediated and cell mediated immune response that will give much more broad spectrum protection against the viral disease. The inactivated Covaxin, since it is an inactivated vaccine, it will not go into the cell, but it will directly bind to the antigen presenting cell, and this will predominantly evoke an antibody mediated immune response with the cell with less amount of cell mediated immune component. So the protection, the spectrum of protection in general in inactivated vaccines is comparatively low than the intracellularly presented antigens. So, but antibody mediated immune response will be equally good for both these vaccines, and these will give us equal amount of protection. But maybe in the scientific sense, in the very strict sense, maybe Covishield will be little better than the Covaxin, but in the practical sense, it may not make much of a difference. Now, the new approaches in the vaccine. So for an ideal vaccine, we would always like to have a vaccine which is thermostable, which can be stored at room temperature rather than having a cold chain. And also without getting an injection, if we can get the vaccine. And this is very important for COVID because this is a mucosal route infection. And we have antibodies in the mucosa, and we have antibodies in the circulation. And the mucosal antibodies are produced by the mucosa associated lymphoid tissue. And there is, these two compartments, the systemic immune response and the mucosa associated immune response, they most of the time they behave independently, even though there is a crosstalk between the two systems. So even if you have a systemic immune response with IgG levels, you may not have sufficient mucosal immune response. That is why at least it is suspected that in some individuals who have got the vaccine against the COVID, they may be susceptible to a second infection. It may not become like a severe infection, but at least the virus can enter and colonize in the pharynx or in the respiratory tract. And when you do a PCR-based testing, you may find it to be positive. And maybe you will have mild symptoms of the disease. but it will not become a complicated disease, because when the virus starts spreading to the system, it will be neutralized by the antibodies. But ideally, to prevent the infection in the beginning itself, it is always good to have a mucosal antibody response also. So now one of the major threshold is to make mucosal vaccine, so that there will be significant amount of mucosal antibody, that is the IgA, and along with that there will be the systemic antibody response, that is the IgG response. So maybe an inhaled vaccine or an intranasal vaccine, and even the SARS-CoV-2 have receptors in the intestine. So maybe an oral administered vaccine may be useful for this COVID. So that is one of the newer approach that is coming, and that will be just like our oral polio vaccine, or maybe like an inhaled vaccine. The newer vaccines are coming up. Now, one of the major, quality control or the approaches used by vaccine manufacturers are like, they strive to develop vaccines that are effective in preventing or reducing severity of infection disease, provide durable long-term protection against the disease, achieve immunity with minimal number of doses, provide the maximum number of antigens that can confer the broadest protection against infection. You would have seen that there are claims by Covaxin manufacturers that their vaccine, since it is the whole virus vaccine, it has, apart from the spike protein, other viral proteins also present, like the N protein present, or the structural proteins of the virus is also present. So this will give a better antibody or better spectrum of antibody response, so that even the new variants, against the new variants of the virus, this will be protected. That is the claim made by the manufacturer, but it is yet to be tested. But there are always possibilities that when we have a broader spectrum of antibodies present induced by the vaccine, that will give us better protection. Now, we always like to have a vaccine which cause no or mild adverse events. Then they are stable at extremes of storage conditions over a prolonged period of time. India being a, we always depend on the and if we can avoid the use of a cold storage, that will be always good. Then available for general use through mass production, and affordable to populations at risk of the infectious diseases. So an ideal vaccine should satisfy these points. Now, there are a large number of immunogens used to develop vaccines, varying from the polysaccharides, conjugated toxoid antigens, subunit vaccines, whole pathogen, just like the inactivated virus, then reassorted live virus vaccines for certain diseases, then potential future concepts. See, essentially, this is an older, or the classical concept of vaccine development, where we were thinking about nucleic acid vaccines and vector-based vaccines as potential future concept. Now, these are the classical vaccines which were available: live attenuated vaccine, inactivated vaccine. Now we have the replicating viral vector and non-replicating viral vector vaccine for different diseases. So this, the non-replicating one, is the Covishield that is being marketed now. Now DNA vaccines are coming up. RNA-based vaccine, the mRNA-based vaccine is already available. Subunit vaccine was already available, especially for hepatitis B, the surface antigen that is being used as a subunit vaccine. So all the newer concept, these vaccines are Newgen vaccines. And the major advantage for having this Newgen vaccine is we don't, for live attenuated vaccine or inactivated vaccine, we need to culture these viruses. And in these particular cases, we don't need to culture the infectious organism. So it itself is a big advantage for vaccine manufacturers because they can have a lower level of biosafety requirement for handling these organs or making the production systems. So at the same time, if it is an inactivated vaccine, also you have to make large quantities of the infectious virus initially, then kill them and make the vaccine for mass use. Now, this COVID is not going to be the last pandemic that the human race is going to face. We have been facing pandemics from very long time back, even from the 430 BC itself. There are cases, documented cases of the the pandemics. And subsequently we have seen large number of pandemics across. And interestingly, for most of these pandemics, we could make vaccines at some point later. Maybe like Ebola, we are still not successful. HIV, we could not make successful vaccines against HIV. But COVID means because of we have this SARS-CoV-2, we had SARS-CoV-2 experience, we could make COVID-19 vaccines very fast. So we were successful also because the pathogen is not mutating that fast. So far we think that the vaccines that we are using is successful. And interestingly, this pandemics most of these pandemics are coming from animals. They are zoonotic infections, and we know that there are ample number of chances for us to get into contact with animals because of so many factors. That is happening. And one of the major reservoir for most of these wildlife viruses, exotic viruses, are bats. And a study recently published, a study has shown that this even in the bats, significant number of new coronaviruses are residing. Around 3,000 or 800 coronaviruses are still present in the bats, which may jump the species and come to human population. There are so many other. These are all different families of viruses, whichever thought to be present in the bats. And any of these viruses can jump to the human species. So there are ample chances of new pandemics coming into the population. So we would depend on vaccines, and we will need newer and newer vaccines in the long run. And as I was telling, there are so many favorable factors for the emergence of pandemic. The evolution of infectious agents, development of resistance to drugs, resistance of vectors to pesticides, then human demographic change, behavioral changes, susceptibility to infection. Immunosuppression is a major reason. Many people are living to a longer, their longevity is increasing. The transplants, especially organ transplants, are increasing. So that needs immunosuppression, which will make the individual susceptible to newer infections. Then poverty and social inequality is increasing at the different parts of the world. Then environmental changes, including the climate change and travel and increase in the travel and commerce, everything is favoring the emergence of pandemic. See, we have seen the SARS-CoV-2, which emerged in the Wuhan, reached India much faster, across the world much faster, and now it has become a global problem. So, if there was no significant travel or commerce across the world, maybe this would never would have become a pandemic of this nature. Maybe 100 years back, if this was happening. So there are ample favorable factors for emergence of pandemic. So we need newer and newer vaccines. And this one of the major thrust is coming on health approach, where we need to look at the environmental health, animal health, and the human health as a single entity. And interestingly, while we were discussing on this One Health approach, there is a new connotation come to One Health in India. We are thinking that Ayurveda, allopathy, and other systems of medicine together forms the One Health approach. That is not the One Health that we need to talk about. And that is not One Health. That is only health about human beings. But One Health is always a conglomeration of human health, animal health, and environmental health in a balanced manner. And we need to look at the interactions or transmissions of disease across these three systems. Now, the vaccines for emerging infections, we need new approaches, definitely. And this COVID pandemic has given us the opportunity. All the technologies which were residing in the laboratory without getting translated into the clinical side got an opportunity to get translated into the clinical side. I still remember 20 years back when I was doing my master's, or I was presenting on DNA vaccines and nucleic acid vaccines, which has been still in the research world, which was never translated to the clinics. And within less than only a time, we see that nucleic acid vaccines have been translated. Even the vector vaccines we were studying and we were teaching about vector vaccines, and now we see that this vector vaccine has taken up a big spectrum in the vaccine market. Now, one of the major advantages of the vector vaccines are we can make multivalent vaccines or multi-pathogen, multi-disease vaccines with the single vectors. We just have to introduce the immunogenic protein coding regions into these vectors, and single injection of one vaccine will prevent multiple diseases. And this offers tremendous potential even for those pathogens which need a very high containment requirement, like a VSL4 facility, like maybe an Ebola virus, maybe a Marburg virus, maybe Nipah virus. We can make vaccines without having such elaborate biosafety requirements. So that is one of the exciting possibilities. And since now these vaccines have been accepted and approved in for the clinical use, I hope that there will be a tremendous boost in the vaccine manufacturing market in the coming years. And we would see a large number of new vaccines coming up, the technology getting much more refined. And this will be one of the major thing that we need to look forward in the coming years. So in the eve of the National Vaccination Day, which commemorates the vaccination or the immunization of first of oral polio vaccine administration in India on March 16, 1995. I hope that our vaccine scenario is going to improve. It is going to better, and we will have more number of better vaccines coming in the days. Thank you.
Thank you very much, sir. Thank you very much for the information and the wise words on the vaccination program and the benefits of vaccination. We will take a few questions from the audience, sir.
Definitely.
We have a question from Mr. George Koshi. George Koshi, can you please tell the question, or should I read it? I think I will read it for you. When can we able to say that a vaccine is successful? The question from Mr. George Koshi is that when can we able to say that a vaccine is successful? 60% or 80% or 100%? And the second part of the question is, do RGCB is associated with any of the vaccine development program connected with COVID-19? And what are the organization ecosystem required for vaccine development?
Okay. So the first question I have already mentioned, like we don't need 100% coverage for any vaccines. We need within the population around, depending on the kind of disease that we are dealing. For example, COVID, we would need around 50 to 60% of herd immunity to give a perceptible change in the disease spread, and it will protect the community. And for individual level, there are yardsticks for making a vaccine, whether that the individual is seroconverted properly and is getting a protection. So there are different assays available at an individual level. For example, if I get a vaccine and when my serum is tested to know whether I am protected, there are certain specific assays to look into. But a community level, around 50 to 60% coverage, it will be sufficient to give a protection. I think I answered the first question. Second question is about RGCB. Right now, we are not into COVID vaccine program. But most of our activities are focusing on antiviral development. And at some point, we may get into the vaccination, COVID vaccine development R&D program. We are not into any manufacturing or into tie-ups with any of the companies who are manufacturing this. But most of our focus is into R&D on what vaccine candidate can be used, what technology can be used, and also like how to improve the immune response against these vaccines. Like how a better adjuvant can be used in a vaccine so that the immune response become better, or the duration of the immunity become better. So at present, we don't have any ongoing programs, but we have some thoughts on how to get into the scenario. We have developed some neutralization assays which can be used to see whether a person who is vaccinated is protected, like a pseudovirion based neutralization assays against a COVID. So this is being developed to look at the vaccine efficacy response. And maybe at some point of time, like depending on how the feedback is, whether we need to modify these vaccines to improve the longevity of protection. These are some of the things that we are thinking about. But at present, right now, we are not into any of the vaccine platforms or programs.
Thank you. Doctor, good evening, doctor. Okay, can you hear me?
Yeah, sure. Yes.
I am Vinod Haridas. I am a management committee member of TMA. I got actually two questions for you, because basically our knowledge is from reading all these newspaper reports. A lot of reports are coming. Now at present, between first dose and second dose, 28 days is the period now they are specific. Some reports are coming that it should be a higher period than 28 for this vaccine to be effective. What is your opinion on that?
I agree with the second thing. I mean, 28 days are. I feel that it is little early. So, a basic concept or a theoretical concept in vaccinology is after giving the first vaccination, there will be a primary immune response, and when the primary immune response is about to wane, I mean, it's about to go away. At that time, we should give the second dose, that is the booster dose. Otherwise, what would happen is the high level of antibodies present because of the primary immune response will neutralize the significant amount of the antigen that you are giving in the second dose. So that is one thing. And also, like, it takes some time for the memory immune cells to mature enough. So that is why we are waiting for a sufficient period. So 28 days are actually, it is on the brim. It is a little early. And there are suggestions that we can even wait up to 3 months. That means from the first day of vaccination, maybe within 90 days, we can have the second dose. Suppose someone personally asks me whether I should stick to that 28 days, I would say even if you wait for 2 weeks or 3 weeks more, that will be good. It will not be bad. So we don't need to 28 days, suppose we are getting a second vaccination date on particular day, we don't have to be too fussy about that second date. But it should not be too much delay, but a few months, like even 1 month delay will not affect much, but that will be good in a way.
Okay, Doctor, one more question. There is a lot of discussion on the effect of alcohol on this vaccine, whether you can take drinks after vaccine. A lot of discussions are going on there. What is the actual position?
Actually, this question has come up because most of the major vaccination programs are always done in pediatric population, and we don't worry about alcoholism related issues in pediatric population. But alcohol in general, especially chronic alcoholism, is in general considered as an immunosuppressive agent. It is not that one day you take little alcohol and that will affect the vaccine response, but it is essentially a chronic alcoholism has got some immunosuppression effect in the body. So that is why in those patients or those individuals, vaccination will not give the optimum response. So ideally, it is not that, maybe it is good that on the day of vaccination or maybe next 3 or 4 days, you don't take alcohol because maybe there is some fever or some tiredness and all that in the system. So it will be always better to avoid that. But I don't think, after a week or so, it will greatly affect the immune response. It's not, unless it is a kind of extreme and chronic alcoholism.
Okay, thank you.
One more question from one of our participants from UAE. The participant is asking, Mr. Vinod is asking, is there any chance to trigger migraine or its occurrence once get vaccinated? The question is that whether migraine will get activated by the vaccination. And is it safe to take vaccination for my son who is below 18? Here the vaccination is given for above 16 in UAE.
Okay, the first part is there are no reports that the vaccination will trigger a migraine. And maybe in most cases, it will be a tension-related headache or anxiety-related headache most of the time. And when as such, no, there are no reports that it will lead to increase in migraine or it will trigger a migraine. So that is based on the scientific information available. But I don't know too much about that, but I don't think that it will precipitate. Second thing is about the age. When he is asking whether he should take vaccine for his son who is less than 18 years.
18 years.
Okay, but the age is 16.
In vaccination, it is 16. In UAE, it is 16 is the age for vaccination.
But his son is already 18.
No. He will become 18 only on May.
Okay.
now he has to face these public exams. Most of the students here, they are giving above 16 years also. So my question is whether 2 months back, I can take him the vaccination now.
I think so. There is no harm in getting the vaccine, even in when he is a very young adult, so he can definitely get the vaccine, no problem. And these vaccines are safe. And I don't think you should refrain from getting the vaccination done. You can get the vaccination done.
Yeah, okay. Because here now Covishield is not, we are not able to opt. Now we have to go for Sinopharm only.
No, I think Sinopharm is like our Covaxin. It is an inactivated vaccine, so it should be working equally good. It will not have an issue.
Okay, thank you, sir. Thank you.
Can I ask a question?
Definitely.
You mentioned that the efficacy rates of the different vaccines are varying. So, if somebody is found to have a low level of antibodies, is it advisable to go in for another type of vaccine?
It's a very good question. Actually, theoretically, even I used to feel it might be good to combine this inactivated vaccine with the vector kind of vaccine, because it operates through different mechanisms. So we will have a broader spectrum of protection. But as of now, all the clinical trials are done with whatever is the primary vaccine given, that is given as the same vaccine is given as the booster dose. So that is the way the clinical trials are done, and the approval has been given. Suppose I want to get first vaccine followed by Covaxin. Maybe for a regulatory authority, they would need another clinical trial report to approve it. So that is the difficulty.
I didn't mean that, doctor. Say after 2 vaccinations with the same type of vaccine, say after 2 months or so, if you take, I mean, again, 2 doses of another vaccine, I mean, will it give a much better level of protection?
It might. Theoretically, theoretically, it might. So for example, if you are taking another vaccine which is working through another mechanism. So one is an inactivated vaccine. Suppose you are taking inactivated vaccine 2 doses, you didn't get sufficient level of protection. Then you thought, okay, let me take a vector-based vaccine for another booster. It might give a better response.
Thank you.
Scientifically, it can give a better protection.
Thank you.
No, any more questions? If there are if there are no questions, then we can proceed ahead with our vote of thanks. I welcome our MC member George Koshi, Mr. George Koshi, for the vote of thanks.
Good evening. Sir, it is very nice to listen Dr. Sreekumar, a long-time associate of mine in KCST, after a brief interlude. So I know that how much pain he is taking in the field of vaccination research. He is a person who is very much committed to the research. And so it is very eye-opening for us from today's speech, we learned very much about how the vaccine programs are conducted and how we can enlighten the people, those who are approaching us with half knowledge or without any knowledge. Because fortunately, I say that we took 1 dose of vaccine, already took 1 dose of vaccine, people coming and asking all sort of questions about how it was like that. So now we are very much educated, and we can tell the people how it is and why it is like that. So your contributions to educate the public as well as the research in this field is very elaborate and very worth mentioning. So on behalf of TMA and my colleagues, I thank you, sir, very much for the time you take out from your busy schedule to enlighten us in this evening. And we hope that this is help all of us, all of our colleagues, to learn much about it and think about it. And so we thank very much for on behalf of the TMA. I also thank all my colleagues in TMA to participate in this evening lecture and contributed their self to the topic, which is very serious topic, because we know that it is discussing extensively and it is a very serious topic. We thank you all who participated in today's meeting, including Mr. Jayachandran, who welcomed us. Thank you, sir.
Welcome. Welcome.
Thank you all. Thank you, Dr. Sreekumar. Thank you all participants. We are winding up the session. So, thank you.